The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You have logged on to the Nelson Labs and RCA webinar, Accelerating Product Release Through RapidCert, patent pending, Biological Indicator Sterility Testing. I just wanted to talk about a few housekeeping items before we get right into this exciting new topic. If you missed one of our webinars or would like to refer back to one, you can always find them on the Nelson Labs website on the on-demand webinars page listed under education. You can receive notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following us on LinkedIn. We also would like to invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Laboratories is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 900 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know that every test matters, to improve and requires solutions to complex problems to improve patient outcomes and minimize client risk. Today, we are joined by Nina Moreno and Jordan Elder. Nina Moreno is a certified microbiologist and global segment director for sterility assurance at Nelson Labs. She has been with the company for over 20 years and has experience in providing technical and strategic oversight for microbiological laboratories. Nina is a member of the Kilmer Microbiological Quality Process and Analytical Technology team and the Modern Microbial Methods Collaboration Group. Jordan has over 15 years of experience navigating regulatory pathways and identifying optimal regulatory strategies for both pharmaceutical and medical device companies. As Regulatory Compliance Associates Director of Regulatory Affairs, he is responsible for providing our clients with general direction, tactical objectives, and functional plans for their regulatory needs. Jordan is meticulous regulatory professional with a comprehensive background in medical device regulations and a demonstrated history of successful product submissions, including FDA 510Ks, EU Class 2A and 2B, Health Canada, and CFDA Class 3 submissions. We do encourage everyone's questions. We will try to get to as many as we possibly can at the end of this presentation, but we do encourage you to submit them at any time in the questions box. And so now, to begin today's presentation, I'd like to turn the time over to Nina and Jordan.
Great, thank you, Mike. And thank you everyone for joining today. We're looking forward to spending some time with you discussing how you can accelerate your product release through this patent pending RapidCert biological indicator sterility test method. So for the agenda today, we will cover these three main items: how the RapidCert BI test complies with expectations for an alternative method, the key benefits of RapidCert BI testing versus the compendial approach, and also the regulatory approach for implementing RapidCert BI testing. Mike just reviewed this information. So here's an image of myself and the other presenter who will be Conducting the second half of the presentation today is Jordan Elder. So as Mike mentioned, please feel free to submit questions. We are certainly happy to help address any questions that you might have during the course of this presentation. You can submit those and we'll answer at the end. So let's start with the expectations for an alternative method. General guidance for the validation of an alternative microbiological method is given in USP chapter 1223. So this chapter covers how to validate and gives some general information in this chapter as well. So first off, in this chapter, there is the acknowledgement that there are limitations to the current methods that are available and are considered compendial. So one example is that the actual limits of detection of compendial microbiological methods have never been established quantitatively, and that the CFU as an estimate with a variety of limitations is prone to underestimate the number of cells that may be present. So I think it's important to first of all recognize that while we feel very comfortable with the compendial method, it is well known and acknowledged that there are certain limitations. So that first example that we just haven't determined the LOD, even though this type of an approach, a compendial approach, is something that we use on a regular basis. And then again, with the CFU limitation, CFU, the colony forming unit, is something that we use regularly in the microbiological setting. And, you know, it's not a direct reflection of the number of cells that might be present within a product due to the way that cells just naturally form together, that they might clump. And so there can be differences in the way that cells are measured versus the way that CFU are presented on a plate. A second item to note from USP chapter 1223 is that there is support for alternative approaches. So it actually says right in this chapter that USP has long provided support for the implementation of alternative assay methods. And this also has a reference to the general notices section of USP, where again it is noted that there is support for alternative approaches. While we might feel comfortable with compendial, it is not the only way to support microbiological test methods. And then the third item to note from USP chapter 1223 is that there are guardrails in place. So the way that the USP is set up is that methods and procedures that are below 1,000 are considered to be referee tests. So if there were ever to be a question in terms of what is appropriate, there is the knowledge that the tests that are below 1,000 are considered the referee tests. And harmonized compendial methods may not automatically qualify the use of an alternative method unless it also meets criteria that might be specific to other compendia, regional requirements. And so while alternative methods can be validated, there are also guardrails in place to protect against any potential issues that could come from using an alternative approach. There are additional references for how to approach and validate an alternative method. I'm going to focus a bit on USP chapter 1223, but there is similar information that's available for the European Pharmacopoeia as well as PDA Technical Report 33. So all of these are great resources for how to approach the validation of an alternative method. So in USP 1223, it gives very detailed guidance in how to approach the validation. So it gives this nice intro section about history and things to consider, and then goes into specific steps on how to validate an alternative method, which this RapidCert BI test would be considered an alternative method. First thing is that the test is considered to be a qualitative test. So there are certain validation parameters that need to be achieved in order to validate this alternative approach when it's a qualitative test. So some examples of a qualitative test would be the sterility test and a test for absence of specified microorganisms. And certainly BI testing falls into that category. It's commonly referred to as a BI sterility test, and we are definitely looking for the presence or absence of a specific organism. In this case, we'll be talking about Bacillus atrophius. The validation parameters that are required to be met as part of an alternative approach are specificity, limit of detection, robustness, repeatability, ruggedness, and equivalency. And so Nelson Labs has completed a validation that covers all of these required parameters. So this is some high-level information for how we achieved meeting those specific parameters. I do want to note that we have additional detailed technical summaries that are available. So if that's something of interest, of course, feel free to reach out. We're happy to work with you on that. The first item is specificity, and this is where items were tested in triplicate for samples that were inoculated with the indicator organism. So again, in this case, that would be Bacillus atrophius. And then we also had a series of samples that were heat shocked. And with this test, essentially what you're looking for is the ability to detect a range of challenge organisms. So in some cases that might be a variety of different types of organisms, but in this case, a range of organisms is the ability to detect Bacillus atrophius in various states. So that's where we use the indicator organism in its kind of inoculated state as well as a heat shocked state. We also validated the parameter for limit of detection. For this, we used multiple replicates at different target inoculation levels, and those were tested, and then the detection probability at each target level was compared to the log transformed inoculum level using a logistic regression to estimate the limit of detection. For robustness, the samples were tested in triplicate for various conditions, for media volume and for temperature. So, you know, the reason for approaching robustness this way is to demonstrate that the test method is unaffected by small but deliberate changes that might happen in the course of the test. So that's what we're establishing with robustness. We also tested for repeatability and ruggedness. For this, we used triplicate samples tested by multiple analysts on different days. We also evaluated multiple pieces of equipment. And in this test, we evaluated the average relative light unit, or RLU. This is the way that the system detects the presence of organisms. And so we evaluated that and compared that to a signal to noise ratio. We also evaluated the coefficient of variation. So we calculated that for each individual test run that was completed. And then we also evaluated that across all test runs that were done. For equivalency, so we used a decision equivalence approach. For this, BIs were processed through a partial kill cycle. We did this for multiple cycles, and we did this for, okay. Some issues here on the screen. Hopefully you're seeing the correct slide now. So for equivalency, we validated for a partial kill cycle, multiple cycles, different BI lots. We made a comparison directly to the compendial method, so the traditional 7-day incubation, to evaluate the equivalency and the non-inferiority of this method. The evaluation was performed using a bootstrap confidence interval. of the difference in detection probabilities with a non-inferiority margin of 0.2 or 20%. And that's what's recommended in USP 1223. And so that's what we followed for this validation. So the conclusion of this validation was that validation acceptance criteria were met and that equivalency or non-inferiority was demonstrated for the Rapid BI test method, which is great. That means that this test method is validated and able to be used and is an approach that is in alignment with the guidance that's given in USP 1223. I also wanted to take a moment to acknowledge the guidance that's given in ISO 11138. So in section 7 of 11138-1, it states that the incubation time and temperature shall be validated. So it's important to pay attention to the use of the word shall. That means that it is a requirement, not a recommendation. So these chapters are specific to biological indicators, and it is required to validate that time and temperature parameter. It's also noted that the reference incubation for Bacillus atrophius is commonly recognized to be 7 days for ethylene oxide, and again, that's what we're most familiar with. But this statement comes along with 2 notes, so this is pulled directly from that ISO 11138-1. The first note states that it is possible to have a validated incubation time that is shorter than the reference incubation time. And then the second note is that national or regional requirements for incubation time validation can also exist. So similar to what I mentioned from USP 1223, that it is acceptable to validate something that's different than a compendial traditional approach, and that it's also important to consider national and regional requirements and ensure that those are being met for the product market that you're working in. The second section of ISO 11338, so this is actually section 7, -7, section 12. It states that manufacturers of BIs are required to provide information on culturing conditions. So this is so that we can follow what's already been established by the manufacturer as an acceptable culture condition approach. So that would be temperature, incubation, media type, and potentially volume. This section also states if the user employs other culturing conditions, these should be validated by the user. So again, it's important to pay attention to the language here. This is where the word should is used, so that means it is a recommendation, and this is what Nelson Labs has already completed for the RapidCert BI test. This section also states that culture conditions recommended by the manufacturer of the biological indicator should be followed. So again, use of the word should. If culture conditions other than those recommended by the manufacturer are employed, they should be validated to determine their effect on the performance of the biological indicator. So again, with the validation that Nelson Labs performed, this is exactly what we were addressing, is the performance of the BI in relation to each of those parameters that were outlined. So let's go ahead now and take a look at the key benefits of the RapidCert BI test versus the compendial method. So it's important for us, if we're going to do this comparison, to start with looking at what the compendial method actually looks like. So here's a process overview for that. The test is conducted by taking the BI from its container, so in this case a glassine envelope, and then placing that into suitable growth media. So this is a soy-based media. That's done in an aseptic manner. That is then incubated for a length of 7 days at 30 to 35 C. After that incubation time requirement has been met, there's a visual inspection of the test containers to determine presence or absence of growth. And so you can see here in this image, there are some containers that look a little bit cloudy. So that usually shows up in the form of turbidity, some type of a film that grows. There can be like a debris or material that's present in the test container. And that visual interpretation and indication is how we decide whether the container is positive or negative for growth. For the biological indicator test, we would then move to some type of an organism identification process to confirm that the organism that is growing in there is actually Bacillus atrophius, that it is the MRO and the indicator organism. That's important because otherwise this could be a sign of potential for contamination in the test. So this is the general process that takes place for the compendial method. So that 7-day incubation, and then several days for potentially an organism identification if that's needed. For the RapidCert BI test, here are some of the key features. So rather than the 7-day incubation, we have validated this to a 48-hour minimum incubation time. So significant savings there that can certainly be impactful when you're looking at your process, how products are released to the market, inventory control. This can be really critical. This can be important for validations and shortening the timeline that it can take to validate the sterilization process for a new product. So many applications where this 48-hour minimum incubation can be incredibly helpful. The second feature is that this is an ATP-based system. So that's adenosine triphosphate. That is a nucleotide that's really only present for viable organisms. And so some of the questions that we sometimes get with an alternative approach is, oh, am I going to potentially pick up the presence of something that's non-viable? For this particular test method that we've validated, that just wouldn't be the case because ATP in these higher levels are only going to be available if the organism is viable. And so this ATP-based system provides objective results that are measurable rather than the subjective use of the visual interpretation looking for turbidity in the test containers. And then the last key feature is that we have validated for 2 common BI types that are inoculated with Bacillus atrophius. So we have options available for different types of BIs that you may already use in your process. Okay, so here's the process overview for the RapidCert BI test method. So you can see it has a very similar flow to that traditional test method. We would take the biological indicator, remove it from its container, still test it in a soy-based media. That would be for a 48-hour minimum time frame rather than that 7 days, and it's still at 30 to 35 C. From there, rather than doing a visual inspection of the containers to look for turbidity, we would remove an aliquot from these test containers and run that through this test system, which allows for the introduction of reagents to detect the presence of ATP, and that is reported out in relative light units. So it is something that is objective, it's measurable, and then that information is interpreted as positive or negative for growth. We still have the ability to perform an organism identification for these tests. So we still have that ability to conduct that organism ID work, confirm that it is actually the indicator organism that is present in the test container and not some other type of potential contamination. For this process, that would include a 48-hour incubation and potentially the need for an organism ID. All of this could potentially be completed within a 7-day time frame. So significant savings there when you're looking at the fact that the compendial traditional approach, 7 days is just going to get you the BI test results and nothing beyond that. It would still take additional time to perform an organism ID. With this shortened incubation, we're able to conduct the organism ID work and that BI test interpretation within the shortened amount of time. So for this test method, one of the main benefits is, of course, that shorter incubation time, that 7 days versus 48 hours, and how this can have a significant impact for your sterilization load and the process that follows from there with getting the product out to market. The second is that there is broad applicability for this validation that's already been performed. As I mentioned, we did already validate for different manufacturers of BIs. We have the ability to validate for additional BI manufacturers if that's something that's needed to support your particular process. And we are also able to validate for different organisms. So if we're looking at something like the need for a geobacillus, that's something that is possible. And I did want to point out that the RapidCert BI test is a great alternative for the self-contained BI. Self-contained BIs do have a reduced incubation, a shortened incubation time, but they can also be potentially prone to breakage that might be difficult to detect within that sterilization load. It can be tricky because an SCBI is not always well suited for the type of environment that it's used in. So this is an industrial environment, forklift, so there is a potential for breakage there. So this RapidCert test is a great alternative for those who are already utilizing SCBIs. This method also allows the determination of results to come from objective instrument-based data. Of course, analysts that perform biological indicator testing are well trained to the process and understand how to visually inspect for presence of growth. But having this be instrument-based does provide that additional level of assurance that we're not going to miss something potentially visually, or if there's something that's difficult to interpret in a visual way, we're able to detect growth faster than we would be able to do by simply just using the human eye. And for this test, we are able to have quick implementation and an easy transition from either a standard BI or an SCBI into using this RapidCert process. Because we've already done the validation approach, that is completed, and there's no additional validation work that would be required in order to start moving to the use of this RapidCert approach. Of course, parametric release and reduced incubation time studies still remain viable options. Those can both either eliminate or reduce the time that's required for test results. But both of those methods do require significant validation work that just wouldn't be necessary if you were going to switch to the RapidCert BI test. So it does support that easy transition. As I pointed out with the visual overview of the process for both compendial and RapidCert, there is broad equivalency to how the test is done. The test is already accredited for conformity to ISO 17025, so if that's something that's necessary for your process, good news, we already have that established, and of course the validation has been done by Nelson Labs, trying to make this as smooth of a transition as possible to be able to start using this RapidCert process. So now I'll go ahead and switch the time over to Jordan to talk about the regulatory approach for implementing RapidCert BI testing.
Thank you. You can move on to the next slide. So first up, I want to talk and take a look at devices that have received FDA clearance under a 510K. These devices are under a lot less scrutiny than a Class 3 PMA device, and therefore have a much lower bar to get to the market with changes. First and foremost, I want to highlight that in this instance, we're going to be discussing devices that have maybe only had one or two other changes, or ideally none, since the originally cleared 510K. We're also assuming that each of these changes would have been minor and not previously required a new 510K. Under this assumption, FDA provides a great guidance document called Deciding When to Submit a 510K for a Change to an Existing Device. And part of that you can see on screen here is a section, it's a flow chart kind of going down to make that determination of, do I need to submit a new 510K? But before we get too far, let's briefly discuss why changes to devices will play an important part as to whether we can use this method outlined by FDA. So when evaluating device changes, FDA considers the totality of changes to the device. This means that a device with one or two minor changes is not likely to impact the overall safety and effectiveness of the device. However, and this is actually a true story, I have seen companies that have also had over 1,200 changes to the device. And the question really becomes at that point, what point in the accumulation of those changes is the device no longer the original device? This is similar to the parable of, if you guys have ever heard of Theseus' ship. And for those familiar with this thought experiment, the real question is, if a seaworthy ship has all its parts replaced slowly over time, at what point is it no longer the original ship? Is it when the mast has been replaced, the planks, the outside of the ship? At what point do we draw that line and say, okay, the ship is a completely different ship than the original? These same principles apply to medical devices. And the question becomes, at what point is this a new device that could actually affect the safety and effectiveness? Unfortunately, with FDA, there is no hard and fast rule. So for you manufacturers, you're going to have to assess this on a case-by-case basis. For here, kind of going down, we can see that we should take a look at chart B based on the technology and engineering performance change here. So if we can move on to the next slide, we see a little bit of chart B and kind of going through. You'll notice that the answer is no, and I apologize, I've had to cut off some of this. This came directly from FDA's guidance document, so I've got the first half here. But you'll notice that as we're going through answering the questions whether Rapid BI Cert could really impact any of these, you're going to notice the answer is no. And it's going to lead us to the same conclusion, which is documentation. So you may be asking, what is documentation? And I apologize. Nina, we can go to the next slide. There are several names for documentation, but the most commonly known technical term is called a letter to file. And that's really just internal documentation used to justify why a change to a previously cleared device doesn't require submission or a new special 510K. Just for you guys to know, FDA doesn't need to clear this documentation, but you do need to have it. You still need to assess and document the changes through internally. And FDA will typically review a lot of documentation such as this on the next on-site audit. So just because they aren't reviewing it now doesn't mean that they don't plan on reviewing it later. And as you can see, some of the critical components, you need to have the description of the change, the reason for your change for switching to Rapid BI, and assessment and justification of the change. And your change can be, hey, we just wanted it done in 2 days instead of 7 days, which is a completely justifiable reason. But you need to discuss and document the assessment of that. So that's going to be updates to things like your risk file, your FMEA analysis, and your risk management plan. A robust description of the change. And you can refer back to the master file that we've submitted to FDA. That's a master file 3939. You need to request a letter of authorization from FDA, and I'll get a little, or from Nelson to provide to FDA, or at least keep that internally. You may want to check on and update your updated lot release acceptance criteria and provide a justification of acceptability and a justification establishing the totality or the entire acceptability of the change. So you may be asking yourself, that's great and all, but those FDA flowcharts looked a little subjective, or if you're familiar with them, they can be subjective. So how can you be positive the documentation will be accepted under a letter to file? Well, the good news is on March 12th, Nelson Labs actually had a meeting with FDA to discuss this approach. And FDA indicated that because there does not impact any validated sterilization methods and only involves adjustments to the incubation period and how we're evaluating this, they essentially agreed that it's not going to require a new 510K. So we can feel pretty confident that this will fall under a letter to file. With that being said, we can take a moment and move on to the next slide, and we'll talk a little bit about PMAs. So like the 510K discussion, FDA indicated during our March 12th meeting that devices will typically fall under a 30-day notice. And what that means is that you provide that notice to FDA, you provide that documentation to FDA, and within 30 days following FDA's receipt of that documentation, and that does require a submission for PMA holders because this is a Class 3 device, as long as the agency doesn't object, you're good to start marketing. So the great news is this isn't going to be a 180-day supplement. It's not going to be a supplement where you have to spend a long period of time waiting for FDA approval. It's pretty quick. Like I said, 30 days. With this, you will definitely 100% have to reference the master file. And like we said, FDA has reviewed this. You may also want to request the validation summary letter from Nelson Labs. And anyone utilizing this method for a PMA, you're also going to want to get a letter of authorization. So this letter of authorization is similar to, Well, I guess to put it in simple terms, it's a permission slip saying Nelson Labs is allowing you to reference their master file, and FDA is okay to use that as a reference when looking through your documentation. It makes it easy, it makes it clean and clear. And just for reference, we are also in process with FDA currently setting up training. So these reviewers who may be looking at your documentation, whether that is a 510K or a PMA, we are setting up a training session with FDA and all applicable interagency personnel, so they are aware and they are familiar with this new Rapid BI process. So that way, when the time comes and they're reviewing it, they don't come back to you with a million questions wanting to understand the science behind it or why it works. They should already be well aware of it, and they will be should be a smoother process moving forward. So Nina, we can move on to the next slide. So with that, I did want to highlight some of the typical documentation included in the 30-day notice. And there is a great FDA guidance document, as I had on the previous slide. But once again, it goes back to, you're going to want a description of that change. You're going to want a summary of the data supporting the change. A statement, and this is a little outside what you would need for a 510K, but the change has been made under the requirements of 520F and 21 CFR 820, which is your quality system. You're going to want that description of your device for a PMA, identification of the manufacturing facilities, a reason for the change, and any appendices of supporting data. The summary of the data or information supporting the change, a lot of this will be found in the master file, but anything else, maybe the procedures established, which Nelson can provide some of that information. Obviously, some of that is also provided in the master file. We've talked about rationale for the sampling method if we plan to verify the changed processes, and a summary of the completed validation study. So a lot more for a PMA. And I will say that most of the PMA requirements probably won't apply to 95% of you. I want to say throughout the entire year, FDA receives maybe 5 or 6 PMAs, and it accounts for a very small amount of all devices that go through FDA, but just things to be aware of as you're moving through. A 510K is relatively simple. You don't need market clearance. You're kind of good to go. Short of having that documentation internally. And I do want to stress that you really do need that documentation before you move forward. I have seen the mistake companies have made where they say, well, we'll get this documentation in place, but we're going to go ahead and make the change and start marketing now. Please don't do that, because you don't know when FDA is going to come, especially if you already have a 510K, that you're marketing. They could show up tomorrow. And if you've started making that change, but you haven't fully justified it and documented it internally, that's an audit finding that I would not want to have from the FDA. So just please be aware, it's not overly onerous to get that documentation in place, but make sure you have it in place before you start marketing with that change. And with that said, Nina, we can move on to the next slide. And if you do have questions, please feel free to reach out. We're happy to help you with whatever kind of documentation, requests or questions you may have. And from there, Nina, I will pass it back over to you.
Okay, great. So here is a QR code, and it takes you to the website that's listed here on the slide. That's a place where you can get additional information about this RapidCert biological indicator test process. And Mike, we're ready to go ahead and take questions.
Great. Thank you both, Nina, Jordan, for this excellent presentation on this new test service. As Nina mentioned, we will go to the Q&A. We'll try to get through as many of these questions as we possibly can before we end our time today. If we don't address your question specifically, the team would be happily address it individually after the event concludes today. So we have several questions that have come in, and we'll just get going. Here is the first question, and again, I'll just throw it out to each of you, and let each, if you want to, Nina or Jordan, either one of you grab it, and then if you want to provide additional comments, feel free. But here's the first question. Is this test able to detect unexpected growth from lab contaminations, or is it specific for Bacillus atrophius?
Yeah, I'll go ahead and take that, Mike. Great question. So this test, you're actually able to detect other organisms as well. So if something else were to be present, that would still show as that particular sample being positive for growth. From there, we would conduct the organism identification process to make sure that it is actually the indicator organism and not some other type of a contaminant. So yes, this test is able to detect a broad range of organisms. So no issues there. We'll still be able to detect it, and then we would do the confirmation similar to the traditional test method to make sure that it is the indicator organism.
Okay, great. We'll go on to the next question. How does the sample aliquot represent the entire growth media contained within the test tube?
Yeah, great question. So because there is this period of time where samples are incubating for that 48-hour minimum time frame, that gives enough time for viable organisms to grow and to be able to be detected using this test system. So while the aliquot is a small portion of the test media overall, we would, of course, go through a vortexing process to make sure that the item is well mixed, and that sampling would be representative of the full test container. Again, because we have that time frame for organisms to go through this growth phase while it's in that 48-hour incubation, we're still able to utilize a small portion of that overall test media and still get great measurements in terms of that relative light unit measurement to make that determination for presence or absence of growth.
Okay, thank you. And Nina, I think these are going to be probably more for you because they are test related. So, but we do welcome your regulatory questions on this as well. The next question is the organism ID included in the RapidCert test procedure?
Yeah, so similar to the compendial traditional approach, the organism ID is only performed when necessary. So if there is growth that's detected in the test system using RapidCert, that's when we would utilize a confirmation of growth. The way that we've typically approached this is if something is detected other than the indicator organism, it's likely that that could have been some form of contamination. And so that would be potentially on the Nelson side. If it is the indicator organism, we're able to perform genetic identification, confirm that it is Bacillus atrophius, and move forward that way. So it's not part of the, I guess, the standard test code for the BI test, but it is part of the process that's built in to confirm that that it is the indicator organism that's actually growing in a test container. Does that make sense? So part of the process, not necessarily built into one test code.
Perfect. Thank you, Nina. Our next question is, what is the risk of contamination by taking an aliquot?
Yeah, no, this is a great question because, of course, we wouldn't want to add to the potential for a false positive through this process. So the good thing here is that because the amount of time from when we're pulling that aliquot to when we're running it through this test system is so short, there isn't time for any potential contamination to propagate. So that would not show up in the test. In addition to that, we would of course conduct this in a controlled environment using aseptic technique. And so the risk of having additional false positives than what you might see in a traditional method just really isn't there because that amount of time to propagate isn't part of the way that the test is conducted. So it's a pretty short time between when we pull that aliquot and run it through the test system. So yes, so that would not get us to the threshold of having it show up as a positive unit if for some reason there were potential contamination during that aliquot process.
Great. Here's our next question. Are those BIs available at each Nelson lab in Europe and China for example. So, where is this test offered?
Yeah, so right now the test is offered in our headquarters in Salt Lake. It will soon be available in Itasca, and we are working on the validation for Wiesbaden as well. So certainly we have plans to expand the locations where this test is available. So more to come there for sure, but we are going through the process of validating the test in different regions. Now the BI itself, just to be clear, this isn't a BI that we're manufacturing. The BIs that we have validated are the large strips for Steris and Mesa. And so we validated the test for both of those traditional BIs that you may already be using. So you can continue to use either of those BI types, and then we could perform the RapidCert on those BIs.
Great, thank you for explaining that, Nina. Our next question is it possible to use in first intention RapidCert BI for process initial validation?
Yes, yeah, great question. So this test can be used both for validation as well as for routine test monitoring. So yes, the way that we approach this validation is we use a 30 to 80% survivor rate. So similar to the kind of process that you might see, for example, in a reduced incubation time study. And so it is appropriate to use this for either a validation or for the routine lot release process.
Okay, here's our next question. And maybe this will be also a for both you and Jordan. So the way I understand how BIs are used, they are included during the sterilization cycle and checked for each batch of med device equipment or med device product. Is the same level of regulatory change paperwork necessary when BIs are used to do initial qualifications of facility sterilizers? Let me know if you need any of that repeated.
Yeah, can you repeat that real quick? I think I know the answer, but I want to make sure that I'm addressing it.
So the way I understand how BIs are used, they are included during the sterilization cycle and checked for each batch of medical device product. Is the same level of regulatory change paperwork necessary when BIs are used to do an initial qualifications of facility sterilizers?
If they're done for the initial qualification, and therefore I'm assuming validation, you wouldn't have to do a change because that's part of your initial qualification and validation. So this is only looking at from a change perspective. If you're going with your new 510K and you're doing this with a new product, that's just all included as part of that initial validation. Please let me know if I didn't answer your question or if I misunderstood.
Thank you, Jinky Jordan. Okay, so our next question is, how would justifying this new incubation method apply to the European community for CE marking a product line?
That's a great question, and it's really going to go back to your notified body and the change control. So it is going to require a change assessment form with your notified body, and a lot of the documentation will still stand as is. The notified body may ask for a little bit more, and that's what Nina kind of indicated. We're preparing some of that. Right now, we've rolled out in the US, but the intent is to roll out in Europe, and with that, we will be working on putting together somewhat of a package that the notified body can use or you can provide to the notified body.
Okay, here's our next question. And these are great questions. Please continue to submit them. What would you estimate for total normal duration for both methods?
Okay, so for the duration of the test itself, the traditional compendial method is that 7-day process, and then the RapidCert shortened incubation time is at 48 hours. So this might depend if you're looking at a validation. So this question is for a validation approach. It may depend on the number of cycles that you're running, because certainly if you have multiple cycles that are going through this process, there can be significant time savings there, as that would continue to add up for every cycle that's being run. So it really depends on the specific situation, but each individual test would still have that 2-day, that 48-hour minimum incubation for the RapidCert.
Okay, so here is our next question. Does the RapidCert BI procedure receive a 510K clearance from the FDA such that it can be used for 510K clearance of a device?
Can you repeat that question?
Yep. So, does the RapidCert BI procedure receive a 510K clearance from the FDA such that it can be used for 510K clearance of a device?
So to answer the first part of that question, no, it doesn't receive its own 510K clearance. We do have a master file with the FDA, and you can reference it. However, you don't need a 510K clearance or really even a master file to provide with the initial 510K clearance for your device. Referencing it should just be enough. But no, to answer the question as a whole, it does not have or need a 510K clearance. FDA wouldn't clear the Rapid BI process itself.
Okay, thank you, Jordan. Here's our next question. For an EO validation half cycle, do you still recommend performing the 7-day incubation in addition to the rapid test for confirmation? Why or why not in accordance with ISO 11135?
Yeah, great question. So for an approach like this, we would still be able to use that RapidCert test method, so that shortened incubation time, and not necessarily try to conduct testing both ways, just because it's simply not required per what's outlined in ISO. So because we have already validated that shortened incubation time, that can be applied both to this type of a half cycle validation approach in addition to the routine processing. So to answer your question, the ISO references would indicate that other incubation times can be used. So yes, we would still be able to use RapidCert for a half cycle validation approach without necessarily also conducting at the same time the 7-day test.
Okay, thank you, Nina. I'm not sure if this is related to that previous question or not, but you quickly discussed validation. Would the use of the Rapid BI require full sterilization cycle validation?
Sorry, Mike, one more time, would you repeat that?
Yes. So you quickly discussed validation. Would the use of the Rapid BI require a full sterilization cycle validation?
Yeah, so the way that we've approached the validation that was done by Nelson Labs is that additional validation work specific to product cycle would not be required. It can be broadly applied to the use of either of the BI types that I mentioned that we have already validated for use. So we wouldn't need any additional validation work for those BI types. We've also done validation work for a couple of our PCDs, so that's PCD 007 and 008. This is where the BI is in direct contact with the PCD material, the tubing and cap, so not contained within a glassine envelope. So we've validated that as well. So all of those should be able to be used with this RapidCert process.
Okay, we do have time for a few more questions. Our next question is, Which type of organism ID would you recommend to confirm the indicator organism? Or would Nelson Labs observe the orange colonies on the growth plate and make a final determination without genetic ID?
Yeah, so if the organism is exhibiting signs of, that indicator organism specifically, of course, with Bacillus atrophius, having that orange type of appearance is certainly a good indicator that it is the indicator organism itself and not some other type of contaminant. But it's important to note that when BIs are run through some of these like partial kill type of cycles, they can actually change their appearance and not necessarily exhibit that traditional orange coloring that might come with Bacillus atrophius normally. And we've actually seen that. So some of these BIs that are run through this like 30 to 80% survivor rate would actually maybe show as like a cream color, something like that. And so that's where the organism identification piece would be particularly useful. We do have a couple of ways that organism IDs are offered at Nelson Labs. So we have the option for a MALDI-TOF as well as a genetic. For this particular case, it's recommended to use a genetic ID approach as that would point more towards a specific strain of bacillus, as opposed to the MALDI, which is also, generally speaking, a great way to perform an organism identification, but can have issues distinguishing between different species of bacillus. And so that's why if we are doing an organism ID, then it would be recommended to do the genetic test.
Okay, thank you. What is the analytical method that is used to detect the ATP in the aliquoted sample?
Yeah, so the way that this is done is basically the ATP that would be present from any organisms that are within the test container would be enhanced through the addition of specific reagents to the test container. And then from there, there's a luminometer in the equipment that would detect that light, that elevated level of light. And that's how we would measure the presence of the organism, is through the relative light units that are evaluated by that luminometer. So this equipment is from Charles River. It's the Celsius equipment. And so that has its own separate validation. It has other applications, but that is the equipment that can detect the presence of organism. And in this case, we've applied that specifically to the indicator organism for Bacillus atrophius.
Great. We'll take one more question, and here it is. Is the capacity at Nelson for this type of testing similar to current capacity for the traditional method, or will queue times be delayed initially?
Oh, great question. Yeah, because if we're touting the time savings that are going to come from this test, we certainly wouldn't want to have any delays. So at Nelson Labs, standard approach is to receive and test samples the day of receipt. That would still be the approach. And then as far as a queue for going through this RapidCert evaluation, so this aliquoting and measuring process, that would also be able to just flow quickly through. So we do have the capacity available to support this test. So this, again, we're targeting that 48-hour incubation and then being able to achieve results right after that. So certainly not looking to add to the time that may be required to get test results. But yeah, great question. And we have prepared on the capacity side.
Great. Well, thank you. Thank you so much to Nina and to Jordan for this presentation and their insights on this very new, exciting capability. We'd like to thank you, the viewer, for attending this session. We hope you found it to be valuable. I do want to note that Nelson Labs does host educational seminars for the medtech professionals to help establish and refresh your fundamental testing knowledge. that will then help you achieve more efficient, accurate, and effective testing outcomes. And by attending today's webinar, we'd like to offer you a discount to this seminar if you visit the Nelson Labs website and register for our next seminar, which will be in Salt Lake City. Please use the code SEMINARS10 for a 10% discount. Again, we thank you for all, and we hope you have a great rest of your day.